Okay, hello. So my name is Brandon Cheslin. Today is May 7th of 2020. And I will be going over my answers for case numbers 2 and 4. So for case number 2, with the MRI, or the brain MRI and the PET scan, the questions were: how does glucose uptake in normal tissue compare to glucose uptake by tumor cells? And the image above, and why would this be advantageous to the invasive tumor? The other question was, what modifications may have occurred in cancer cells to alter glucose uptake as predicted above? So, I'll start with glucose uptake by cells happens at a slow rate in normal tissue. Carbohydrate metabolism starts with digestion in the small intestine, which is absorbed into the bloodstream. Blood glucose levels are then controlled by insulin and epinephrine, and cancer cells can multiply under many conditions and metabolize glucose at a higher rate, and that higher rate is known as the Crabtree effect. So with the Crabtree effect, it's observed that an increased glucose uptake by cancer cells decreased their oxygen consumption. So it appeared to be a requirement to divide and proliferate rapidly and to increase anaerobic glycolysis in cancer cells. Let's see. So modifications. I asked what modifications may have occurred to alter the glucose uptake. So the Warburg, I think I said that right, Warburg phenomenon, and the p53 gene mutation. So for the, my dog's in the background doing some weird stuff. The Warburg phenomenon, it was discovered that due to damage in the mitochondria of cancer cells, the cells go through anaerobic glycolysis. And the cancer cells can produce energy in the presence and in the absence, but it is preferred in the absence. So that implies that the cancer cells must adapt to the increased glucose in order to produce enough energy. So for the p53 gene mutation, the p53 gene is a protein that suppresses tumors. It regulates cell division, keeps a cell from dividing too fast or without control. And it promotes a mutation in the gene promotes tumors. A mutation in the p53 gene also allows the cancer cells to proliferate uncontrollably. So, also it will not allow death, which can cause the division of a damaged cell in the mutated p53 gene. So, whenever the tumor cell is considered, then the glucose uptake for those cells increases as the cells must perform the continuous cell division that requires the energy source for the event to proceed. In regard to the tumor cells, the glycolysis rate is much higher and must get ATP using glycolysis as well as the Krebs cycle, and last but not least, with the ETS pathway. PET imaging shows that the majority of the tumor has a higher fluorescence because of the glucose level in that part of the brain is much higher. However, in the regular brain cells, the glucose is needed in small portions to fulfill processes and needs. When we talk about the liver, it has two major roles with the glucose. On one note, extra glucose can be stored as glycogen, means the other glucose amount is actually used for energy production and glycolysis in regard to multiple functions. As most of us are probably aware, as far as kidneys go, when the urine contains glucose, that is a pretty good indication of diabetes being present. Along with the fact that glucose level in the kidneys have been elevated, which is a good indication of diabetes as well. So, on one more aspect of cancer or tumor cells is that these cells make the new blood vessels around themselves to get more food material for their growth through the process of angiogenesis. Again, I think I said that right. These vessels can also help them to provide with the glucose, as they are the leaky vessels. As far as the report goes, I think the cancer tumor cells would need a higher amount of glucose as contrasted to the cells that are merely surrounding them. These cells are having the damaged DNA, thus they have the high probability to undergo the apoptosis. Thus, to increase the population, soon this glucose is used as the energy source. Please excuse my dog in the background. So for case number 4, let me get back to wherever it's at. As a previously healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports she has not given the baby anything to eat other than breast milk since birth. The baby has only recently been sleeping for more than 2 hours between feedings, and that's when the mother reports these symptoms started. Lab results are consistent with hypoglycemia, but otherwise unremarkable. Epinephrine was administered. There's no increase in blood glucose, but there is an increase in blood lactate. Physical exam reveals a liver edge 4 centimeters below the right costal margin. Percussion of the right chest and abdomen confirms enlarged liver. Well, yeah, we'll go with the words that are in parentheses. Blood glucose levels increase after breastfeeding, but she cannot maintain blood glucose levels within a normal range during fasting. So the main question, based on the hereditary, I was reading question A on this. Based on the information below, which of the following is the most likely diagnosis of this patient? So we have A, hereditary fructose intolerance, B, galactosemia, C, glycogen storage disease, D, glucose 6-phosphate dehydrogenase deficiency. So for number 4, I believe the baby is suffering from glycogen storage disease. The baby's symptoms is enlarged liver, which the baby was showing, low blood sugar level, which the baby was also showing that's part of the symptoms, and a high level of lactate. The baby was not showing like fats and uric acid in the blood, at least it was not reported, and impaired growth and delayed puberty, bone thinning, increased ulcers, mouth ulcers. That's what the baby was not showing. So with the symptoms that were present, they're all getting matched with glycogen storage disease. So as far as the others, why they were kind of ruled out, just very short and brief here. For the galactosemia, the symptoms would be like loss of appetite and vomiting, which was not present The hereditary fructose intolerance, I don't believe that was it. Again, nausea, vomiting, gas, abdominal pain, fatigue, and malabsorption of certain nutrients. Again, that was not shown in the symptoms. So for the deficiency, let's see, the symptoms would be dark urine, yellow skin and eyes, and pain in the abdomen and the back. So I would say based on the symptoms that were present and symptoms of the other options, they weren't lining up. So I think the best option would be C, the glycogen storage disease. And epinephrine raises glucose levels in the blood. It plays a role in the fight or flight mode by supplying blood to muscles, stimulating the release of lactate. For the given condition, there is not an increase in the glucose level, but the blood lactate level increases. In glycogen storage diseases, there is a deficiency of the enzyme glucose-6-phosphatase, which is an enzyme that is found mainly in the liver and the kidneys and plays an important role in providing glucose during starving conditions. As the infant is unable to maintain a blood glucose level during fasting states, this indicates that there is something defective in the enzyme function. This enzyme cleaves the glycogen to glucose and leads to hypoglycemia, low blood sugar, and lactate formation, conditions indicating that baby is unable to maintain sugar level but produces lactate due to the epinephrine. The physical symptoms would indicate a swollen and inflamed belly due to the enlarged liver. And I have provided my sources in the script attached, but I hope everyone enjoyed the video. Thanks.